Welcome to this explainer. Okay, today we're diving into the fascinating, highly structured biological blueprint of how your kidneys filter metabolic waste while maintaining perfect fluid homeostasis. If you're studying advanced biology, whether that's for A-levels or your board exams, you already know the kidney isn't just a simple filter, right? It's a highly calibrated chemical processing plant. So, in this explainer, we're going to break down the exact mechanisms, track those pathways, and highlight the highest yield exam concepts you absolutely need to master. Let's get right into it.
So, let's kick things off with a massive number just to set the scale of what we're dealing with here. Every single day, your kidneys filter a staggering 180 liters of fluid. To put that into perspective, that's the equivalent of about 90 large soda bottles of fluid rushing through your internal filters daily. That is huge. Yet, miraculously, less than 1% of that is actually excreted from the body as urine. The rest, it's carefully reclaimed. So, how on earth does the body pull off this massive recycling effort?
Well, the engine behind this whole recycling operation is the nephron, your kidney's microscopic functional unit. A healthy adult has about 1 to 1.5 million of these in each kidney. Blood comes in, it gets filtered, and then it's processed. But before we get into the microscopic weeds, you absolutely must memorize this golden mathematical equation. Excretion equals filtration minus reabsorption plus secretion. Seriously, write that down. Everything we discussed today is going to plug directly into this golden formula.
Now, before we look at the process itself, let's quickly contrast the two main structural players in the kidney. We've got two different lengths of nephrons, and they have very different capacities. First up, the short cortical nephrons. Think of these as your standard, everyday workers. Second, you have the long juxtamedullary nephrons. These are your deep-sea divers. They dive way down deep into the renal medulla, and that specific structural length is exactly what allows your kidney to deeply concentrate your urine when you're dehydrated.
Okay, so how does that single drop of fluid actually transform? We're going to track it through three definitive chronological stages. Stage 1 is glomerular filtration. That's where the initial sorting happens. Stage 2 is selective reabsorption, where the body frantically grabs back exactly what it needs. And finally, stage 3 is tubular secretion, which is the active removal of any remaining toxins. Let's walk through this timeline step by step.
All right, stage 1. This basically acts like a high-pressure sieve right at the renal corpuscle. Blood flows into the glomerulus through the afferent arteriole. And notice the physics at play here. The efferent arteriole, where the blood actually exits, is narrower than the afferent arteriole entering it. This size difference acts like a bottleneck, building up a capillary hydrostatic pressure of about 55 millimeters of mercury, or 7.3 kilopascals. This pressure physically forces water and small solutes, things like glucose, amino acids, and urea, out of the blood, across the basement membrane, and straight into Bowman's capsule.
But wait, right here, I need to point out our first high-yield corner. This is a classic exam trap that catches students literally every single year. Please note, ultrafiltration is an entirely passive process. It's driven solely by that intracapillary blood pressure we just mentioned. That means absolutely no ATP, no active cellular energy whatsoever is spent during this first stage of filtration. It relies entirely on cardiovascular hemodynamics.
Moving right along to stage 2. The filtrate enters the proximal convoluted tubule, or PCT. Now, if the glomerulus is a passive sieve, the PCT is the aggressive recycler. It does the heavy lifting. This section selectively reabsorbs about 67% of the filtered water, sodium, chloride, and potassium. But even more critically, it achieves a 100% recovery rate for glucose and amino acids. Under normal physiological conditions, absolutely none of these valuable nutrients should make it past this point.
So how do these solutes actually get back into the body? Well, they have two distinct routes. They can take the transcellular path, moving directly through the interior of the tubular epithelial cells, which often requires spending some active energy. Or they can take the paracellular path. This is where they passively slip through the tight junctions between the cells, kind of like sneaking through an alleyway between two buildings, just following their concentration gradient.
The absolute engine driving this energy-dependent transcellular transport is the sodium-potassium ATPase pump, which is located on the basolateral membrane of the tubule cells. These cells are absolutely packed with mitochondria to produce ATP, and they burn that ATP to actively pump sodium out of the cell and into the interstitium, pushing it right against its concentration gradient.
And why is that pump such a big deal? Well, think of sodium as the lead domino in a massive biological chain reaction. By actively pumping sodium out, the tubule creates a lumen negative potential and these massive concentration differences.
Once that first sodium domino falls, it passively drags chloride, water, and even urea right along with it. The water essentially just follows the salt through osmosis.
But you know, the kidney is incredibly smart. It actually self-regulates. What happens if this flow is moving way too fast or sodium levels get too high? Enter the macula densa. This is a specialized cluster of sensor cells sitting in the distal tubule. If excessive sodium reaches this point, the macula densa cells literally swell up with water. This physical swelling triggers them to release a paracrine signal, adenosine. This adenosine then smartly constricts that afferent arteriole, reducing the blood flow and dropping the glomerular filtration rate right back to normal levels. It's a brilliant built-in feedback loop.
Next up, the filtrate drops into the loop of Henle, named after the German anatomist Friedrich Henle. And guys, this isn't just a transport tube, it's an incredible countercurrent multiplier system. As the filtrate drops deeper down the loop, it creates a massive concentration gradient in the tissue fluid of the kidney's medulla. We're talking up to 1,200 or 1,400 milliosmoles deep at the bottom curve. This hypertonic, super salty environment is the absolute key to recovering water later on.
To really picture how this multiplier works, let's use an analogy. The loop of Henle has two distinct sides with totally opposite rules. Think of the descending limb as a water slide. It's highly permeable to water, allowing it to escape passively into that salty medulla, but it completely blocks salt. Conversely, the ascending limb is like a powerful escalator. It actively pumps out salt, sodium and chloride into the medulla, but it is totally impermeable to water. Pumping out the salt on the way up is exactly what draws out the water on the way down.
All right, after surviving the loop, we arrive at our final major process. Stage 3, tubular secretion. If reabsorption was all about saving the good stuff, secretion acts as a highly targeted biological trash chute. Here, any remaining toxins, excess potassium, creatinine, and hydrogen ions are actively pumped from the peritubular capillaries directly into the tubule. This lets the body eliminate harmful substances even faster than simple filtration would allow.
Okay, time for another quick high-yield corner. I want to highlight this critical exam distinction because the terminology trips people up all the time. Do not confuse secretion with excretion. Secretion is the active, microscopic transport of waste from the blood directly into the kidney tubule. Excretion, on the other hand, is the macroscopic, final elimination of urine from your body down the urinary tract. They sound kind of similar, but biologically, they are distinct milestones in that golden equation we started with. Now, in the distal convoluted tubule and the collecting ducts, the bulk of the work is already done. Here, the endocrine system takes over the final fine-tuning just to maintain perfect homeostasis. It's highly regulated at this stage. Let's break down this hormonal fine-tuning. Aldosterone steps in to trigger more sodium reabsorption and potassium secretion. Parathyroid hormone signals the reabsorption of calcium. An atrial natriuretic peptide, or ANP, acts as a counterbalance. It actually forces the secretion of sodium if blood volume gets a bit too high.
Let's wrap this explainer up with a quick 10-second rapid-fire summary. If you take away just 4 bullet points today, make it these. 1. Glomerular filtration is purely passive, driven by hydrostatic pressure. 2. The proximal tubule uses ATP to save 100% of your glucose and amino acids. 3. The loop of Henle uses ascending and descending limbs to multiply the medullary concentration gradient. And 4. Secretion is the active dumping of toxins into the filtrate.
I want to leave you with a slightly provocative thought. Considering the massive 180 liters of fluid we filter daily, relying entirely on that hypertonic medulla to reabsorb 99% of our water, how quickly do you think our entire cardiovascular system would just crash, running completely out of fluid, if those energy-dependent salt pumps in the ascending loop of Henle just suddenly stopped working? Think about the fragility of that perfect biological math. Keep exploring, keep questioning, and use these visual blueprints to completely master the biology.